I have the fortune of being able to work with amazing students year after year.
Introduce yourself.
Oh, I'm Suzuki. I am an assistant professor at JCDI. I'm working with John. So today I want to tell you what these students are doing. So these are creative and skilled students, as evidenced by Emmanuel here, doing high throughput biology, closing 2 Eppendorf tubes at once. And in addition, sometimes focus is needed to solve problems, and these individuals here contributed greatly to the project. So for me, the excitement about minimal cell is this. These are 3 different model organisms in yeast. Systematic deletions study found that 80% of the genes are non-essential, or they cause non-lethal phenotypes. 60% of them had, 60% of the genome had normal growth. And similarly, in E. coli, 90% are non-lethal, and vast majority of these show no normal growth. So you disturb these cells and nothing happens. These numbers are much smaller in our minimal cell. Based on the transposon bombardment study of the SIM3 cell in Clyde's paper, I think 25% may be non-lethal, and only 8%, maybe only 8% shows normal growth. So you do anything, well, you knock out genes in these organisms and you're almost guaranteed to get a phenotype. So in yeast, it's not that very few genes are involved in viability. Systematic double knockout studies show that 80% of non-lethals have synergistic lethal pair. And that says that redundancy is masking the basic machinery of life in these organisms, and that machinery is exposed to us in a minimal cell. And this makes it a wonderful platform to study life. So to understand biology, one approach is to inactivate genes and observe phenotypes to characterize the genes. That approach is a little challenging in our cell because clean knockout will result in lethality for most of the genes, and you have no material to look at. So you need a conditional inactivation of genes. Then you can make biomass with at the permissive condition, and then activate or inactivate genes, and then study the population with lots of cells. Traditionally, temperature-sensitive mutants were used for this, and these days we will use inducible CRISPRi. So TET regulation would be nice to drive CRISPRi. I don't know how to, I wonder if you guys also see the icons. Yeah, we tried to implement this and it was not easy for us. But when we implemented a system developed for M. genitalium, we had a breakthrough. And as you see, we succeeded in getting 40 to 60-fold induction of M-cherry here. It's shown maybe over here. And once you can drive M-cherry, you can drive DCAS9 to achieve CRISPRi. So this was done here. CRISPRi, by the way, is a way to target nuclease defective Cas9 to a DNA site to cause transcriptional inactivation. Here, when M-Cherry was targeted now with CRISPRi, we achieved in greater than 80% reduction in expression, as you see here. And it's not that just that the M-Cherry can be targeted. We can target endogenous genes as well. Here we selected 3 quasi-essential genes and 3 essential genes, and found that CRISPRi reduced growth for essential genes here. And then when the growth phenotype showed that maybe CRISPR is not working for these, but when we looked at the transcript level, the expression was reduced by roughly 95% here. So this opened the door for developing CRISPRi tools for the rest of the genome. At this point, we published a paper, and then that was a high point for our group. So this is our scheme for constructing CRISPRi constructs. PCR is used to introduce DNA barcode to enable mass experiments, and PCR is used to add gene-specific guide RNA sequence. And then this small construct we make here is now amplified and combined with a plasmid backbone that provides dCas9 and CRILOX for targeting to the landing pad of SIM3, and then also PureR SIM3 or SIM3B. And we do the mycoplasma transformation, and then pick colonies, and we genotype using PCR and Sanger sequencing. Here's one example where 3 different barcodes are observed, plus guide RNA sequence specific for that target. There are a few clever features in this scheme. The first is to create this construct in a small plasmid and then combine with a big backbone. We learned the hard way that PCR introduces errors, and when we were just adding guide RNA sequences into a big plasmid, we had a big setback. But right now, we're using the backbone that's cloned in E. coli and sequence verified. And then in this final sequence run to determine the guide RNA and the DNA barcode sequences, the whole region that's amplified with PCR is covered. So this results in the error-free construction. Okay, so where are we now? We started with 455 guide RNA or PCR, priming pairs, and then we have now have mycoplasma strains for 102 of them. So 1 clone established for this many, and then 2 clones established for this many, and so on. So maybe 102 is a nice number, but this looks like maybe almost like a start of the project. But this gray area has lots of stories. So procedural, challenges could be one. We have, E. coli steps, PCR steps that can fail, and many constructs drop out. but also there's a biological reason too in this. For example, some of the genes are so sensitive to inactivation, even in the uninduced state. The small effect of CRISPRi kills the cells. Then we would not be able to recover strains for those guide RNA. So for us, the next challenge will be to, I guess, completely understand the procedural problems that are sort of a new target, and then bring these numbers up to that, and then we will be able to I guess leave just the biological problem more interesting problem for the future study. And 102 genes targeted is already quite good. So this year we like to start using these clones. And Shige, participating here, is offering to do transcriptomics on these strains. So we would like to collaborate with them again to make that happen. I guess the first thing we would like to see is that the targets are, repressed with CRISPRi, and we want to see what other genes are up or down regulated, and that will start to help us define the genes of unknown function in these cells. That's it from me. I'll happily take any questions.
Thank you, Yo. What fraction of your 102 are essential versus quasi-essential or non-essential?
Oh, a very good question. We have to, I have to see that. Okay. Very good question.
Hello, Yo. That's a great job. Like always. So do you, there was this previous talks about mutations. If you would propagate those things induced, or like, or do you, even if they are not induced, do you see that they're getting mutated, those cassettes that you insert into mycoplasma? Would they lose the function quickly? If you continue propagating them?
Yeah, we don't have a experiment to tell you anything. Let me see. Yeah, I wonder about the time span of the inactivation studies we do. I guess you see lethality and very little propagation there. We keep incubating that and say get growth. Something probably happens.
Yeah, I just wonder, as you continue study later for all kind of different functions, right? Because I guess the goal is to use it for many different, once you have your clones, to continue, subjecting them to all kind of different pressures, and I guess whatever the assay is, but how stable they're gonna be over time, over generations, or would be kind of maybe interesting to know.
Yeah. Okay. Good point. And we'll investigate that.
Okay.
Anyone else this morning? Yo, thank you very much.
Thank you, John. Thank you, everyone.
I see we still have 63 of you here. Thank you for hanging out for the whole 4 hours and 30 minutes. I was really, really impressed with what I've heard today. One of the comments in the chat is some of you are presenting data that is unpublished, and I'm going to write each of you who presented today to ask for your permission to post your presentation. And if you want, I think we can arrange to have it password protected. Shige Kakazawa suggested that it only be available to people who attended the workshop. So we can arrange that also, but I will send a note around to I think it's 19 of you who presented today, and then I'll do the same next week. But thank you for participating. I have learned a lot. I hope you all have, and I hope you've gotten just a sense of, how large our community is and how it's growing and how many papers we're about to see over the next year or so. So with that, I officially declare session 1 of the Minimal Cell Workshop closed, and I look forward to seeing you all next week. Thank you very much. Any closing comments from anyone before I say goodbye?
Many thanks, John. Great initiative.
Thank you. Thank you, Olivia.
That was really great. Thanks, John.
Thank you very much, all of you.
Thank you a lot, John. It's good to see that there are like that many people working with the minimal cell.
It is. It's super.
I'm alone. I'm less alone now that Donnie came back. In a way, we're alone here in the South Hemisphere, but that's good.
Yeah, indeed, but I mean, sounds great. Hi, Yoko, Bogomil. Great to see you all.
Bogomil, I hope we can get the minimal cell declared
Olivia, great to see you.
one so you can have it.
Yes, I mean I am super interested that you are bringing this up. So because you wouldn't believe it, but actually we start working on ecoliplasma here now a little bit. You're always gonna say like, are you crazy? But there are some groups. There are a few groups actually that we reach out. They are, I don't know if you are working with any of them, but they were finally successfully transforming ecoliplasma with some transposons. And then we want to develop the whole genome transplantation. But again, I would love to start working with the minimal cell, and because of the safety reasons.
Hopefully we can get this taken care of.
Yeah.
Yo, good to see you, Yo. Okay, all the very best. See you next Monday then.
Thank you.
See you.
Amazing.
See you, Olivia.